the screen now. If at any point people can't see anything that I'm doing, please just pop up and let me know. But to begin, I'd just like to mention that largely what Ricardo's group, who I work with, do is try and create blood-based predictors of various human characteristics and disease risk. So today I'd like to talk about how we create estimators or predictors of various human traits using a variety of omics data from blood. I'd then like to talk about why it might be interesting or important to try and predict the levels of different proteins in our blood. And then lastly, talk about the translational element of our research, in which we're making an app called Detector that allows people to view estimated values for various traits of theirs, such as smoking status and body mass index, and compare this to a reference population. But to begin, you might ask, why would we turn to blood to try and create predictors of human traits? And firstly, blood can give us a good overall impression of the health status of our body. And while tissue-specific biomarkers are always going to be advantageous or desirable, blood tissue is well placed to allow us to create predictors of disease in other tissues, such as renal disease and neurological disease. And I'm not sure about any of you, but I'd much rather have my health profile estimated from a single blood draw rather than somebody come along and try and take a sample of my pancreatic tissue or my brain tissue. And in addition to disease risk profiling, you can simply just recapitulate information about someone's metrics, such as their age, height, or body composition from a single blood draw. So how do we actually create these predictors? Say if we wanted to create, for example, a predictor of someone's height, we could do this. In order to do this, we could look at genetics. And we would look at genetics because it's commonly used and also now widely commercialized in the form of genetic testing and genetic health profiling. So if we wanted to predict somebody's height, we could simply just look at, or we could measure the heights of various individuals, and we could look at their genetics to try and identify mutations that are responsible for causing these height differences. In reality, these mutations are just single base changes, also known as single nucleotide polymorphisms, or SNPs. And we would use this information to create a genetic score or proxy for height. In order to calculate the score, we could simply look at, we'll say, 100,000 sites across the genome. And we might find 5 sites where people's genotypes correlate strongly with their height. In reality, this would be much more. Bear in mind that at each site, people will either have 0, 1, or 2 copies of the height-altering mutation. If we want to predict height for person X, we could simply look at each SNP in turn and simply count the number of copies this person has, each height-altering mutation. In this example, all SNPs increase height for simplicity. But if one were to decrease height, we could simply just add a minus sign in for the SNP. Now you might imagine the scenario where some SNPs increase height more than others. To account for this, we could simply rank the SNPs in terms of how influential they are in relation to height. So we could give the most influential SNP the greatest weighting and the second most influential SNP the second greatest weighting, and so on. And we would simply just adjust our scores accordingly. In practice, these weights would come from association studies or other statistical or machine learning methods. So to calculate the score, we just adjust these calculations, and if anyone spots any errors, please forever hold your peace. Summing all this together gives us a genetic score of 11 for height. Now, to give this meaning, we could simply compare this against a range of genetic scores against the population. and this person in particular would be in the 30th to 40th percentile. Or we could apply some form of data transformation that returns this back to a value of height. And that's how we create predictors. This can be applied to any trait, continuous or discrete, and can be applied to other types of omics data. In our lab, we focus on epigenetic data. And for those who would like a refresher, if you imagine that your genetics serve as an instructional manual for your body, epigenetics would change the way or help the way your body reads this code. In our lab, we focus on DNA methylation, which is an epigenetic modification where methyl groups are added to DNA, and this affects how the gene is interpreted. In effect, it acts like punctuation in the code. To illustrate this, if you took the sentence, "Let's eat, Granny," and you took away this comma, it would very much change the meaning of the words, even though they're all the same. In many ways, methyl groups also have this effect at the level of genes. When methyl groups are added to DNA, they change the way they're interpreted and affect gene expression. These methyl groups typically occur at sites which are called CPG sites. This is where a cytosine follows a guanine. Typically, DNA methylation can silence the expression of genes. This often occurs if CPG sites are added at promoter regions, and this is the start site of gene expression. But if methylation occurs later on in the gene, such as in the gene body, this can have the effect of increasing gene expression, so the picture isn't totally clear. This is also a highly dynamic process and is specific to various cells and tissues. Before, where I mentioned in the case of genetic data that we might have 0, 1, or 2 copies of each mutation, methylation data can typically be reported in values of 0 to 100%. For example, here, if we said a CPG site had 50% methylation, what we mean is that 50% of cells in the sample would show methylation at that site. So to calculate a predictor of height for methylation, we would apply the same principle as we did before. We'd look at a number of CPG sites across the genome, depending on which array you use. And we might again find 5 sites where this time methylation levels correlate strongly with height. And we apply the same principle as we did before. So we look at each CPG site in turn. We look at how methylated it is across the sample and adjust our scores by our weights and sum them all together. So this would give us a methylation-based score of 8. But do these predictors actually work? In a previous paper, we calculated genetic predictors shown in blue and epigenetic predictors shown in green for a variety of human traits. And this was done in 2 Scottish cohorts, Generation Scotland and the Lothian Birth cohort. And some predictors worked quite well. So here, a methylation-based predictor for smoking could explain 60% of variation in smoking behavior. People are even trying to make epigenetic predictors of death. Liu et al. Last year made an epigenetic predictor of mortality, specifically from methylation data, and they called this grim age because higher values meant grim news. We then decided to calculate grim age in 900 individuals, all aged 70, in the Lothian Birth cohort. After 12 years of follow-up, we found that those with the oldest grim age, or highest values, had a survival probability of just 50% after 12 years, whereas those in the bottom quartile, or the 25% with the youngest grim age, had a 95% survival probability over the same time period. To me, it's quite terrifying that you can get this separation of prediction from just a single blood draw at age 70. So just as a reminder, you would take some form of biological data, pop this into a statistical method or machine learning pipeline, and the output is a predictor of a human trait. These biological data can be MRI scans, they could be questionnaire data, or in the case of our lab, we use blood data. Specifically, we use genetic and epigenetic data, in part because these regulate the levels of different proteins. The reason why this is interesting is because differences in people's blood protein levels have been linked to various disease states and can even serve as predictive biomarkers. But what we don't understand is how these differences in blood protein levels occur. Therefore, an aim of our lab is to try and identify SNPs and CpG sites that might regulate the levels of these proteins. Not only would this help us predict the levels of different blood proteins, but it also might inform protein biology and also link proteins to disease. We applied this to a panel of 92 neurology-related proteins in the Lothian birth cohort, and we found a number of SNPs and CpG sites that were associated with blood protein levels. A major application of this research, as I mentioned, is that it can link proteins to disease. You might know that a SNP in gene X is associated with Alzheimer's disease, but you might not understand the mechanism by which this occurs. You then might find that the same SNP increases the levels of its protein. This might lead you to speculate that this is the mechanism by which it's mediating disease risk. We studied this in relation to an immune protein called poliovirus receptor, and we found that a previously known association between a SNP and Alzheimer's disease risk, where we found that the same SNP increased levels of PVR levels. This led us to speculate that maybe increased PVR levels were associated with Alzheimer's disease risk. We used a causal inference method called Mendelian randomization, which suggested evidence that increased PVR levels were indeed associated with increased Alzheimer's disease risk. We tested the converse, but it was not likely that this was true, suggesting that this was a unidirectional relationship. This research can also help you identify protein pathways. We identified a SNP in the Siglec-9 gene that was associated with increased Siglec-9 levels. This in itself may not seem very interesting, and it's a cis-acting effect because it occurs in the same gene. But we also found a SNP in the ST3-Gal6 gene, which was also associated with increased Siglec-9 levels. This is clinically important because in a blood cancer called multiple myeloma, increased levels of Siglec-9 help cancer cells evade detection by the immune system. In the same cells, increased levels of ST3-Gal6 have been shown to be upregulated in these cancer cells and help promote Siglec-9 function. And our data help support evidence for this co-regulation and may point towards a mechanism underpinning it. However, there's a limitation of all these studies that I've mentioned, and that is that they try and correlate each SNP or CpG site with the trait of interest in isolation, and they correlate them with the trait of interest just one at a time. The problem with this is that different SNPs are going to be correlated with each other because they're inherited together. Different CpG sites are also going to be correlated. For example, smoking might affect many different sites in the same way. And genetic and epigenetic data are of course not going to be mutually exclusive either. For example, if you lack a cytosine, this will destroy a CpG site, and epigenetic changes can even induce mutations. And together, not accounting for these can lead to inaccurate reporting or estimation of effect sizes. To counteract this, Matthew Robinson's group in Lausanne created a software called Bazar Plus. And what this software allows you to do is associate SNPs or CpG sites with your trait of interest. And while doing so, it accounts for all other genetic factors, all other epigenetic factors, and hidden confounders. I was able to spend some time with this group, and in addition to meeting my childhood hero, we also studied the genetic and epigenetic architecture of 70 inflammatory proteins. Just to briefly mention how this method works, it's a form of Bayesian penalized regression. And to break these words down, penalized regression means that some form of penalty is applied. Here, our penalty is that most SNPs or CpG sites will have no effect or correlation with the trait. And then the Bayesian portion, we assume some prior beliefs. These are updated by the data. Our prior beliefs are that we assume that most of the markers will have some small effect, some will have a medium effect, and a small amount will have a very large effect. And here, a large effect might mean that a SNP accounts for 10% of variation in your trait. Importantly, this software can tell you how much variation in your trait is attributable to genetics, methylation, and the two combined, but while conditioning on each other, and the breakdown of how much is attributable to small, medium, and large effects. Just as an example, we applied this to two proteins, and for one protein, FGF19, we found that it was more so driven by genetics in green as opposed to epigenetics in gold. The converse was true for VEGF-A, and in fact, over 60% of variation in VEGF-A levels could be explained by genetics and epigenetics combined. This was the most for any protein that we studied. So together, this information gives you very important information in terms of the molecular regulation of different protein levels. We're going to apply the same approach to a larger panel of 4,000 proteins in around 1,000 individuals who are members of a Generation Scotland sub-study called Straddle, and this aims to stratify resilience and depression longitudinally. We'll identify SNPs and CpG sites that associate with the different blood protein levels. And as we did with the other predictors, we'll use this information to try and predict protein levels. We'll do this in a separate 10,000 individuals who did not have proteomic data measured. We'll then test whether our predicted protein levels can associate with health outcomes, and that will help satisfy one of our goals of trying to integrate omics data to predict human traits and human health. I'll now move on to the translational element of our research, which is our app methyl detector. So this focuses on methylation data, and the user would upload some form of methylation data. It can have missingness, it can be in any form, and our software will automatically correct this and generate a set of scores for various traits for you. So it will give people scores of their body mass index, alcohol consumption, smoking status, etc. And I'll give you an interactive demonstration of this in a moment. But just firstly to note that in a separate app, you would upload the data and the output is a file ready for use by Methyl Detector. And just to show you how this works, this is the app itself. So you would upload your data. And the first thing that it will do is it can give you the predicted age based off of methylation data for any individual in your data set. You can then select one of our predictors. So here I've selected alcohol, or predicted alcohol consumption. And here in the dotted line, it shows the individual that you've selected. This will then compare their percentile against the input data set, the distribution which is shown in pink, and also our reference population, Generation Scotland, shown in blue. It also gives you here the average score for those who identify as light, medium, or heavy drinkers based off of a Generation Scotland questionnaire. And these buttons here allow you to change the comparison to males and females, and also different age ranges as well. And the percentiles will change accordingly, as well as the numbers in your data set. You can apply this to any of our predictors and for any individual in your data set. And you might see that the percentile here changes for each. You can also just simply look at a percentile plot that shows you your percentile against Generation Scotland for all of the traits at once. And you can also download these ranks here for all of the individuals in your input data set. As well as this, we just have a little bit of information here for people to refer to. And this shows the correlation between all of the different predictors in the input data set and Generation Scotland. And again, this updates as you change your reference characteristics. And lastly, this panel just shows you how well each of the traits correlate with their epigenetic predictors. So here, how well the predicted scores for alcohol correlate with actual alcohol consumption in Generation Scotland. And again, this updates based on the characteristics you choose. And in essence, that is how Methyl Detector works. A future direction of our research is that we're going to add in other predictors from ourselves and our collaborators, and we will ensure data privacy and confidentiality, both in terms of when the user uploads data and also in terms of the Generation Scotland data that we've generated. We're going to apply a similar study design in predicting protein levels in prevent dementia, and this is another Edinburgh-based cohort that's run by Craig Ritchie. For example, we can try and predict the levels of cerebrospinal fluid proteins, see if we can proxy this from blood. And lastly, our group will continue to refine methods for creating predictors. I'd just like to finish by thanking all of the following people, and thank you all for inviting me along to the Journal Club and for your attention.
Great, thanks Robert for a great talk.
Thank you.
So does anyone have any questions?
Hello.
Hi.
Hi. Sorry, this is going to sound like maybe a stupid question, but when you're saying you're predicting traits, and sorry if you did explain this and I missed it, do you think that this is also going to be something that you could predict things like cognition, or risk for, like, obviously you're looking at risk for dementia, but you're focusing more on the neuropathology. Are there plans to also look at cognitive scores within Generation Scotland or these other?
Yeah, so you could try and predict, performance on cognitive tests, and to do this, you could just take kind of scores of people's performance on different cognitive tests, generate the predictors as we did for height, body mass index, and whatever. So you can apply this to anything really, and also pop that in Methyl Detector, but we haven't done this yet in my group.
Okay.
But it's a trivial thing just to pop in if somebody else has done it or plans to do it, it's easy for us to just pop it in and incorporate it.
Okay, cool. Thank you.
Hi Rob, thanks for a really good presentation. Can you hear me okay?
Yeah.
Yeah, I was just wondering, do you see if there is like an application for something like Methyl Detector to be applied to at-risk groups, so like using it to identify, individuals maybe before, like, clinical symptoms manifest fully. So if you could use it to identify at-risk groups and then kind of use that as a way of targeting intervention and streamlining those people for early intervention.
So I suppose, in theory, you could, you might have some part of the app that kind of spits out a line that kind of suggests that the average score here is a cutoff for, I don't know, maybe depression risk. So like it might be a depression risk score, and here these are the people who might be at risk or high risk, low risk, in the same way that you kind of had like light, medium, heavy drinkers. But the problem with all these methylation-based predictors is obviously they're not going to be 100% accurate. And here we've included ones that have reasonable correlation with their traits of interest, so maybe like over a 35–40% correlation with the trait of interest, which at an individual level might not be so useful. So at an individual level, it could be very much missing the actual phenotype, but at the population level, it can be correlating quite well. So I think in terms of moving forward for clinical biomarkers, it probably doesn't meet the standards in terms of not being kind of specific enough or sensitive enough at individual levels. But I think just kind of as an information at the population level, it can be more useful in that sense to kind of demonstrate currently the state of the art, what these predictors can do. So I think in theory, it can be simply applied, but probably not maybe so well applied to something like kind of clinical cutoffs, because, or for something like a disease, because it just won't be accurate enough.
Cool, thanks.
Sorry, just first of all, really interesting stuff. It's very impressive, that app. Do you think it would be like, so just kind of following on from what Neve was saying, so at the moment, would you say that it's more of like it's more of a tool for research and or basic research in terms of like sort of discovery of maybe new like biological pathways or stuff, or what would you say is the application of it at the moment?
Yes, I think just kind of the words that you used there were perfect for it. So I think at best at the moment it's probably a tool for research, as opposed to something that could be applied clinically, or the kind of goal of a lot of these methylation-based predictors is maybe that they can supplement other kind of gold standard forms of clinical management and assessment. And I think it's moving towards that as the predictors get more refined and more accurate. But I think mainly what it can do is it kind of draws on a previous paper that we did where we looked at epigenetic prediction of traits. And I think sometimes people have difficulty in applying actually using the predictors that we generated in that paper to maybe look at the distribution of values in their own data set. So I think what it can do is, yeah, it's a tool for the researchers and maybe just makes that a little bit more accessible.
Cool.
And kind of at a population level, adds a little bit of interest in terms of what these predictors can do. But yeah, I don't see it as something that's clinically useful at the moment.
Okay. All right. Thank you. So, can I follow up with another question? Is that all right?
Yeah.
Do you think it will be at some point? Like, I know this is very in the future and stuff, but what do you think it would take, just out of interest, for it to become more of a clinical tool?
I think as long as it showed very high sensitivity and specificity, like you could maybe just consider the case of one individual predictor or methylation-based predictor or genetic predictor. And I think there are some out there that correlate very well with their specific trait of interest. In the case of all the ones that I showed in methyl detector, smoking is quite well, it does quite well in terms of predicting smoking behavior, and that's because smoking affects methylation quite strongly. So they can perform very well or be quite close to kind of fully accurate in terms of predicting the trait of interest. But as far as I'm aware, none of them are there for a clinical trait quite just yet to be sensitive enough to be approved by something like the European Medicines Agency or the FDA. So I don't see them as being like an approved clinical biomarker yet. But I think people are working on various Bayesian approaches that are generating more accurate predictors and different machine learning pipelines. So I could see it as being the case that it could be, with increased sample sizes, more people getting methylation measured, and kind of improved or refined statistical pipelines, but at the moment, no.
Okay. All right. Thank you very much.
Hello, it's
Hi.
Jen.
Hi.
Really interesting talk. So when I first saw the title, what I would imagine about multi-omics, I would say that genotype and then methylation profile, they may have sort of parallel effects on types, rather than methylation being a mediator of genotype to behavioral traits. And I wonder if, but from your presentation, it seems more like the latter interpretation. And I just wonder if there's any trait that you find genotype, I mean, PRS predictors and methylation predictors have more of an additive effects rather than effects that is lying in the way of, I don't know, one is the mediator of another. That's the first question. And then the second is a bit nerdy. I just wonder, how did you write that app? Is it in Python or?
So for the app, we wrote it using Shiny.
Okay.
With great difficulty, but we got there. And for the first portion of the question, yeah, I think what you're getting at is very important, in that I think it's very important for people to try and understand, if you're using genetic and epigenetic predictors, what's the actual underlying architecture is. Are they completely overlapping? Are they happening in a pathway, or is there some form of additive effect? So I think for the predictors of protein levels, you would have the case of kind of SNP going to CPG site, going to protein level, because that's kind of makes sense in terms of biological pathway. And that's kind of what we see, is that they're occurring in the same region. So the kind of genetic proxies that we have are kind of directly influencing the epigenetic proxies that we have. So they're kind of not necessarily in parallel. But we find that for some of our predictors, if I just go back to one of the slides, sorry, better to go back a bit. So for the human traits, we see here that we just simply here in orange had a combined epigenetic and genetic predictor. And we find that these have independent and additive effects. So if they were completely overlapping, you might imagine here that they just kind of absorb into each other and you wouldn't get any more predictive power over the epigenetic trait once you combine in the genetic one. So it seems to be for kind of complex traits you're getting additive and independent contributions, but I think for the protein levels, it would be the case that they're completely overlapping in terms of their information.
Thank you. That's very interesting. Do you think it's possible to just test, since you mentioned complex traits, maybe the more polygenic the trait is, maybe the effect may appear to be a bit more additive? Well, it's just a guess, but
yeah, I think it would be quite easy to test that, I think, with the BASR Plus platform that's kind of being developed mostly at the moment, because it really helps kind of whittle down the various contributions that you're having in terms of the genetic effects, epigenetic effects. So I think that would help us understand the architecture behind the traits a little bit better. But it's not really something we've looked at yet, and I can see your point there. I think it's an important consideration.
Thank you.
Okay, so if no one else has any comments or questions, I'll just stop